Thank you, organizers, for giving me this opportunity to speak on this symposium. I really very excited about this. And today I'll give you really overview of what is going on in the field of immunotherapy, the way we are heading to and what we are up to by using some of my data that is focused on melanoma, but that can be applied to the other type of cancers. So I'll try to cover three different areas. One is to give overview of adopted cell therapy for those colleagues who are not directly involved in the field of immunotherapy. And then I will go over what we've been working at Repertoire Immune Medicines and end my talk with some of my preclinical data to convince you that when we use interleukin 12 in a form that is tethered on the surface of the cell, it performs better than its soluble form. So adopted cell therapy is really a treatment where we use patient's immune system to get rid of the cancer. And the reason behind this is that the nature has equipped us with an excellent system where the immune system can not only recognize the damaged and infected cell, but can also take care of the cancerous cells, which has been originated from the certain tissues. There are three major approaches that have been going on in the field. One is called TIL, tumor-infiltrated lymphocyte. And the second approach is the T-cell therapy, T-cell receptor therapy. The third is the CAR-T, chimeric antigen receptor T. Each of these approaches has their background, how they come into the field. For example, in case of TIL therapy, and patient don't have enough cells, or even if they have enough cells, these cells are not active enough to take care of the cancer. So in order to keep these cells in enough quantity and to keep them active, clinicians take those T cells from the tumor of the patient, expand them and put back into the patient so that they can take care of the tumor. The way T cell work, they have to recognize a specific marker that is expressed on the target cells. We, in scientific language, called antigens. So in order to have that antigens recognized by the T cell, they should be expressed on the tumor cells or the target cells. Unfortunately, sometimes the patient's T cells are unable to recognize those markers. So we have to introduce those specific receptors into these cells by using genetic approach so that the receptor can recognize that particular antigen and then take care of the tumor. The limitation of these two approaches is that really their antigen has to be bound to a complex protein, which is called major histocompatibility complex. And that sort of make their efficiency limited. In order to overcome this limitation, scientists have introduced another approach, which is called CAR-T. And this is one of the best known adopted cell therapy approach in the field. So in case of CAR-T, really the T cells are not bound, that the antigen of interest is bound through MHC, rather they can target those cancer cells where antigens are not bound to MHC. So in other words, they have more flexibility to target more number of cancer cells than the top two approaches. So post a couple of years, there's been a lot going on in this particular area of interest. You can see from here, the CAR-T is a winner clearly, and even within 1 year, there's a tremendous development in the field of science, starting from the preclinical development different phases of clinical development. And some of these moieties have even got into the market and really performing well in case of hematological malignancy, but still there are issues with the solid tumor. You can see along with the CAR-T, TCR and TIL therapies are also moving forward slowly, but the therapy that we are working is a bit unique among all these, and that will fall into this category. Our approach is really like a little different. We use autologous approach where we get lymphocytes from the patient blood, and we enrich them lymphocyte into T cells and monocytes. And the monocytes, we convert them into dendritic cell by using a cocktail of different cytokines under well-established protocols. And then since dendritic cells are the best known antigen-presenting cells, we present multiple tumor-associated antigens to those dendritic cells. And then the T cells will be trained by these dendritic cells. So in other words, when we will put back these T cells into the patient, these T cells will be able to recognize those tumor-associated antigens into the patient and try to eliminate them. Along with this, we are also trying to tackle the tumor immunosuppressive environment by using different cytokines and immune modulator. And three well-known immune modulator we have in the field is interleukin 15, which is well known to keep T cell in their young form, and interleukin 12, which activate both innate and adaptive immune system. And the third is toll-like receptors, which has been reported to play an important role in the antigen presenting to the T cells. So the data that I'll share you is about interleukin 12, and I'll tell you why we call it deep in next slide or so. So interleukin 12 is really well reported in the field because it activates not only the endogenous immune system, but also adaptive immune system. The way it do is that it secretes a cytokines called interferon gamma, and then interferon gamma perform anti-tumor activity by using different modes. For example, it can increase the level of major histocompatibility complex 1 on the surface of the cancer cell and make them more visible to the T cells so that the T cell can recognize those cancer cells and get rid of those cancer cells. Other is that it sort of convert immunosuppressive macrophages into more like anti-tumor macrophages, and interferon gamma has also been reported to secrete different chemokines. And some of these chemokines are well reported in anti-angiogenic properties and the recruitment of T cells in the core of the tumor. However, interleukin 12 has not made into the market, and there is a reason for that. And the reason is the toxicity. There is a well-reported study from Rosenberg lab here at National Cancer Institute in USA, where they use conventional tumor-infiltrating lymphocyte approach to get rid or to treat a melanoma patient. But you can see rather than the regression of the tumor sort of progress. And then Rosenberg and colleagues use those T cells did the genetic engineering and make them to secrete interleukin 12, and then use those TIL which were secreting interleukin 12. And by using this approach, really, they completely eradicate the tumor with less number of cells that has been reported with the conventional therapy. And the sad story about this clinical trial is that there was toxicity issue due to which the clinical trial has to be discontinued. So nonetheless, there is a message from this study is that interleukin 12 holds strong promise for cancer immunotherapy. So we as a company thought there is a potential for the cytokines to deliver in the field what if we overcome the toxicity concern, and then we will be able to make it to the market and the patient can benefit from it. So the way we try to address this is that we use this approach where we tether IL-12 on the surface of a fragment of antibody. In this particular case is a CD45 antibody. So the third is that because CD45 is expressed on almost all the lymphocytes. So when we will put this construct, which we name as deep IL-12, on to the T cell, it will bind to the CD45 antibody that is expressed on T cells. And we have shown very comprehensively that we can control the loading of this construct deep IL-12 on T cells. And what it means is that we can control the activity of IL-12, and that will minimize the toxicity issue that we have seen with this cytokines. For this purpose, we use this model, preclinical model, female B16 melanoma model. Female is the transgenic mice, and the cell that we got from this female mice will be referred as female now onward in this talk. The specialty about this mice is that the T cell receptor from this mice has specific affinity against a specific antigen, which we name it GP100, and this antigen is only expressed in mouse melanoma cell. So when the mouse has melanoma with this particular cell, when we inject these cells into these mice, it will go to the tumor and eliminate the tumor. In order to do that, we have to do multiple steps. We have to isolate T cells from the mice, expand them, activate them so that we have plenty of cells to play around, and then load our target construct on these T cells so that it will also address the immunosuppressive environment inside the tumor. So by using this approach, we have shown that when we did cell, which we refer as female here, and the cell which were tethered with deep IL-12 below here with multiple dosing, either one mean the single injection was done, and the second and third mean multiple injections were done, just to encapsulate what we will be doing with the patient. So there is a clear benefit that in compared to the control, when we use cells only, we see a little bit regression of the tumor, but by using the multiple dosing, we see really good regression of the tumor over the period of time. And that also prolongs the life of the animals compared to the cell that we only use here, emphasizing that this is a good way to go against melanoma. Next, we want to really see where these cells that we injected in the mice were able to make into the tumor or not. So we take the tumor out from the mice and see the number of these adopted cells that we put into the mice. And we see that there is not only the presence of these cells, but also the expansion, emphasizing that indeed deep IL-12 molecule is expanding these cells and keeping them active also, because all of the adopted cells that we see in the tumor of the mice, they were positive for interferon gamma, emphasizing that indeed the tumor regression is happening through the interferon gamma secretion inside the tumor. But at the same time, we really don't see any activity in the spleen by using this model. You can see none of these adopted cells into the spleen are positive for interferon gamma, emphasizing that we don't get non-specific toxicity with this approach. Next, we want to see is our approach contributing to the endogenous immune system of the mice or not. So we took the tumor from the mice out and take the monocytic myeloid deprived suppressor cells from the tumor to see if there is any activation or is there any negative effect going on. So we look for different markers. We see the highest level of PDL1, programmed death ligand 1 expression on those myeloid deprived blood cells, emphasizing that if there is a high level of interferon PDL1 on these cells, it might be compromising the efficacy of our product, which is DPIL-12 loaded PML. So in order to address this concern, we use combination therapy that is very common nowadays in the field, where we use checkpoint inhibitor, antibody against PDL1, along with this, our traditional therapy to see if it can improve the survival of the animals or not. So you can see here, compared to the control animals or the cell-treated animals, even our product alone, when we use a combination, it prolongs the survival of the animals even with single dose. By using multiple dose, it even increases the survival of these animals to more than 100, which, keeping in view the size of the animals, it is very really encouraging in the field. So overall, what I have shown you today is that by using this tethering approach, we can come over the limitations of interleukin 12, which is the toxicity issue, and we see durable regression of the established tumor, particularly in this model, by using biological approach instead of using genetic engineering. And the combination with checkpoint really further improve the efficacy. And we are very hopeful that this molecule will go into the clinic and do benefit the patient in the long run. And with that, I will like to thank all my colleagues and helper who helped with this study. And thank you for very much for this opportunity again, and looking forward for any question if any of you